As we have discussed in our previous modules about the strain improvement for industrial exploitations through random mutagenesis as well as site-directed mutagenesis. As concerns the site-directed mutagenesis, there is modern technique which we call a recombinant DNA methodology, in which we add some desired gene into a new organism, and there are so many expression vectors, and we can utilize the machinery of some microorganisms, in which the Escherichia coli and one yeast, Pichia pastorius, is very common to use. So when we develop any mutant, then the next step is the mutant selection. So the whole strain improvement and the whole mutation strategy totally based upon the mutant selection. If we are not strong in mutant selection, then our whole process of mutation will go into very usefulness lines. So in order to select any mutant, so when we say that following exposure to the mutagens, when any organism is exposed to any mutagen, the cells should be suitably diluted and plated out to yield 50 to 100 colonies per plate. As I have already told you, when we are dealing with the random mutagenesis, we have to first produce the kill curve, and we have to select that point in which there is only 1% survivors, or we can say that more than 90% kill. So at that level, there is a very limited number of colonies which we get on the plate. So the selection of the mutant is greatly facilitated by relying on the morphology of the mutant or on some selectivity in the medium. Sometimes that the morphology of the organism may change, and then by comparing with the parent means in which there is no mutation. So either there is a change in their medium. So when morphological mutants are selected, it is in the hope that the desired mutation is pileotropic. What is pileotropic? Pileotropic is the mutation in which the change in one property is linked with the mutation in another character. So the classical example of the pileotropic mutation is to be seen in the development of penicillin yielding strain of Penicillium chrysogenum. It was found in the early days of the development work on penicillin production that after the irradiation strain of Penicillium chrysogenum with smaller colonies, and which also sporulated poorly, were better producers of penicillin. So these are the changes occur in that mutant, because that have a very smaller colonies and having a very poorly sporulated. So as concerned the isolation and the selection of auxotrophic mutants by the replica plating methods, as we have seen the difference between prototroph and auxotroph. So when we develop the mutants, there are two types. One is auxotrophic mutant and others are prototrophic mutant. As concerned the auxotrophic mutant, if they have lost the ability to produce that metabolite which is necessary for their growth. So as concerned the prototrophs, they have all those essential metabolites which are used for their growth. So as concerned the auxotrophic mutants, so by replica plating method, so mutants can be detected by selecting or testing for an altered phenotype. So the positive selection involves the selection of the mutant cells and the rejection of mutated cells, which we called as almost parent cells, to identify the mutant that have gained some physical trait. So the replica plating is used for negative selection. Negative selection identifies the mutants that have lost some physical trait. For example, the auxotroph that have the nutritional requirement not possessed by the parent cell. Ames test is a relatively inexpensive and rapid test for identifying the possible chemical carcinogens. So this test assumes that the mutant cells can revert to the normal cells in the presence of the mutagens, and that many mutagens are the carcinogens. So if we add that carcinogenic agent into the medium, then it assumes that mutant cell will revert back to the normal cells. As concerned the histidine auxotroph require histidine in the medium of Salmonella are exposed to an enzymatically treated potential carcinogens, and the reversion to the non-mutant state are selected. Before considering the method used for the selection of method producing improved level of primary metabolites, it is necessary to study the mechanism of control of their biosynthesis, such that the blueprint may be drawn accurately. So if we already know the production potential of the mutant and then the parent, then we have to compare with that mutant. So by the actual quantification of the product and then we comparison in all terms, then we can have a maximum selection of that mutant, which we called as the positive mutant, for better productivity.